Hello and welcome to Murphy Microbiology. I am Dr. Laura Murphy, and today I'm going to be demoing a technique known as the isolation streak. Hopefully you've had a little bit of background already in microbiology and aseptic technique. So that being said, there are some equipment that you're going to have to get together before we can start this particular protocol. One, of course, is going to be the Bunsen burner. We're also going to be using auger plates. There's different types of media you can use for this technique. This happens to be a TSA plate. You're going to want to have some sort of bacterial culture for isolation. You can have that culture either in a liquid form, or you can have it in a solid, on a slant, or on a plate. You're going to want to have some sort of marking implement that you can use to actually label and mark up your auger plate. And then lastly, your transfer tool. I'm going to be using the loop today. You can see this is a wire that has a little bit of a loop on the end. These I find to be the best for doing really good isolation streaks. So let's go ahead and get started. As I mentioned, you always want to start off by labeling your plate, and your instructor will probably tell you different ways that they prefer to have you label. I prefer to have my students label it along the outside edge with initials, the date, and the organism. At this point, you're going to want to give yourself some guidelines for doing the type of isolation streak that we're going to work on today, which is known as a T-streak isolation or a 3-section isolation. There are other methods out there. Some people prefer to do a quadrant streak method, but I find that for my general microbiology students, this one is the easiest to master, and it gives you the best results in the classes that we do. So to start off, we're going to just draw some general guidelines onto the plate using your Sharpie, forming a T. And really what this is doing is just creating the sections that you're going to use as guidelines for your streak moving forward. Having done that, we're going to start off by selecting our organism for the isolation. Today I'm going to be using the broth culture that I have here. This happens to be E. coli, which is a very common organism that we use in the lab for this type of protocol. If you haven't had any microbiology before, you may wonder what I'm doing right now. This is a very important part of any technique that we use with microbes, which is the process of sterilizing our transfer tool. We sterilize just by moving the transfer implement back and forth through the flame until the wire gets red. These are specially designed to withstand high heat, and in doing so, we're incinerating any contaminants that might be on the surface. After I have heated that, I'm going to pass my test tube through the flame a couple of times and then gently insert the loop into the culture. I'm now ready to begin the isolation streak technique. So, starting in the top corner, which is the smallest section of the streak plate, I'm going to gently place my loop and drag it back and forth across the entire surface of the plate. Place it down, and then very importantly, sterilize my loop. This step is often missed by beginning microbiology students, and it causes problems with isolation because you'll end up having too many bacteria on the loop and never really getting pure culture in the end. Okay? So once I have sterilized it and make sure that my loop turns bright red, I'm going to let it cool down. Another important step so that we don't actually kill or burn the organism. And then I'm going to rotate the plate so that my second section is up at the top. Okay? I'm going to test my loop by tapping it on the auger. It seems cool enough. And then drag my loop into that first section 2, maybe 3 times, and then just back and forth across the remainder of that section. Place the plate down and flame again. Okay? And then we just repeat it one more time. I've moved it so that my clean third section is at the top. I start in the top left corner, drag into that second section 2 or 3 times, and then back and forth. And now you have successfully completed an isolation streak. The goal here is to do essentially a dilution of the bacteria, starting from the most concentrated section to lesser and lesser concentrated sections on the plate, so that by that third section, we actually get isolated colonies when you come back after incubation.